Alright, so this is part one of our wrap up of the prokaryote bacteria, archaebacteria lecture from yesterday. This should be about where we picked up last time, and so what we're going to start off with here is talking about differences between different types of prokaryotes. We have the domain bacteria on one hand, with a wide variety of incredibly common organisms that are all around us and inside of us, and really can't get away from them. And then we have archaebacteria, which we tend not to know quite as much about because they're, for one, very difficult to grow in a lab setting, and number two, very often live in very extreme environments, places that are hard for us to get to study them. There are some differences between them. We've talked about things before about how uh, prokaryotes and eukaryotes are different. Looking at different types of prokaryotes now, we find differences in the lipids in prokaryotes, bacteria versus archaebacteria. We find differences in prokaryotic cell membranes, archaean versus bacterial again, ester bonds in one versus glycerol skeletons in the other one. Um, One of the other things that we find that we talk about a lot with prokaryotes, we've heard this term before, is peptidoglycan. And so peptidoglycan is a substance found only in the membranes of bacteria, but not archaebacteria. It's something that is exclusive to the domain and kingdom bacteria. Nobody else has this. So it's another feature that we find fairly commonly in those organisms that is one of the reasons why they've been as successful as they are. We've talked about DNA replication before, as well as cell division in bacteria in general. One thing that we find between eukarya and archaea is that the DNA replication is actually relatively similar, at least as compared to traditional bacterial type replication mode. Additionally, transcription and translation look a lot more like eukaryotic gene expression than it does bacterial gene expression. So those features together are generally considered to be evidence for the fact that archaeans and eukaryotes are more closely related to each other than either archaeans or bacteria or archaeans in any other group. Right? So we have this similarity, this sort of a side group between the archaea and the eukarya. We know these organisms are incredibly abundant. We'll talk more about this number later on. We also know that we don't have anywhere near even a close approximation to good information about most of them. We don't even know a lot of species that are out there. They can obviously live in any kind of environment, in any kind of organism, in any type of habitat, anywhere where you can possibly have life. Bacteria are going to be incredibly common there. So they're always single-celled, they're always unicellular, typically going to be smaller than about 50 or so microns, usually around like 10 microns or less is probably a more realistic figure, so they're very small. We know they have a single circular chromosome, and we know that they have very little in the way of junk DNA, unlike our own genome, which has a lot of intervening sequences of either DNA that we don't fully understand, or DNA that is perhaps regulatory, or DNA that maybe has no function at all. Maybe it's leftover relics from past infection by the retroviruses that we've mentioned a few times before in the past. Reproduction, since they are indeed single-celled, clearly then they cannot produce sperm cells or egg cells, and so therefore they cannot reproduce sexually. That being said, they do have ways of exchanging genes. A little bit later on, we're going to talk about a different type of horizontal gene transfer as opposed to vertical gene transfer, which would be like passing genes on from parent to offspring in the process of perhaps mating with someone else, combining some genes, creating some diversity. While bacteria cannot do that, they do have some other very interesting ways of exchanging their genes in and among their populations. They also have very little in the way of internal compartmentalization. We know, for example, that they don't have much in the way of organelles. They have ribosomes. That's really about it. They have nothing in terms of membrane-bound organelles, so no endoplasmic reticulum or Golgi apparatus or anything like that. So very little in the way of internal compartmentalization. We also talk about how bacteria make a living, and we've seen some of these terms before. We've talked about autotrophs and heterotrophs before. Photoautotrophs, for example, would be your sort of average run-of-the-mill plant. It uses sunlight, that's the photo part of the word. It's also autotrophic, meaning it gets its carbon from a non-living, inorganic type source. So photoautotrophs are sunlight-powered, self-feeding, generating or getting carbon from an inorganic source type organisms. Chemoautotrophs are going to be organisms that get their carbon from, again, an inorganic, non-living type source, but rather than using sunlight, they're going to generate their energy to gather the carbon through some type of chemical means. So perhaps oxidize some other type of chemical, some other extreme environment type bacteria can use some other types of molecules to generate energy, which can then be used to fix carbon. Photoheterotrophs are probably the least common of the four types here. Photoheterotrophs get their energy from light, and they also get their carbon from some sort of organic or living source. Really not too many of those out there, but in the bacterial world there are some. And then finally chemoheterotrophs, which would be where animals are classified. So we're getting our energy from a molecule of some kind. We're not getting it from sunlight. And getting the carbon from an organic or living, or at least previously living source. And again, we find bacteria in all four of these categories. Cell division is very, very fast. We've talked before about how bacteria under good conditions can create a generation in 18 or 20 minutes. E. coli bacteria, for example, is fairly well known to be able to do that. Using binary fission, the fact that these have relative levels of simplicity, they're able to divide very, very quickly. And because they can divide so quickly, that means they can also evolve very quickly, right? So before, when we talked about evolution, we talked about how gene pools change over the course of time. An individual organism cannot change its gene frequencies. An individual organism cannot, by definition, evolve. You have to have time, and you have to have many, many generations in a row. Well, since bacteria can produce so many generations in such a short period of time, they can adapt and perhaps gain new adaptations and spread those adaptations much more quickly than something that has a longer generation time. So that would also be to their advantage. Okay. So in the last chapter, we talked about how we classify viruses. We're going to do the same thing now and talk about how bacteria are classified. Initially, sort of in the old days, the old school version of microbiology, we would look at staining properties. So you guys have seen a gram stain now in lab. Perhaps colony morphology, which was something we unfortunately didn't get to see very much variation. Most of our colonies were fairly homogeneous, sort of a cream-colored, more or less round, shiny type of colony. But there are lots of other types of colonies that can be rough edges or dome-shaped or different colors, yellow or pink or orange or those kinds of things, depending on the type of bacteria. Once someone found that a bacteria stained a certain way, then they would start looking at whether or not it was photosynthetic, whether or not it was able to move, whether it was more or less unicellular on its own by itself, or if it liked to occur in colonies or filaments or groups of some kind, the sort of how the different bacteria aggregate together once they're in a petri dish, what they look like in their natural state. Are they able to form endospores? Are they able to divide a particular way? And then finally, whether or not they were pathogens. And so all of these things are fairly qualitative types of ways of classifying bacteria. Somebody brings you bacteria, you're a microbiologist 50 years ago, you would look at these types of things. Fairly broad, fairly coarse, fairly qualitative, still fairly objective, but nothing really quantitative here necessarily so much. So more recently, we've gotten into more of the molecular classification of bacteria. So now, if someone discovers a new bacteria, they might look at the sequence of amino acids for a certain protein. Bacteria that are of a certain species or of a certain type or strain will have proteins that are all more or less the same thing. And if they differ by a lot, then you might say that they're not part of that group. We can look at the sequence of DNA. How much guanine and cytosine do they have? We can look at does their DNA hybridize well with other strains or other species? And if it does, there's a good chance they're either the same species as the others or at least very closely related to those other species. We can look at gene sequencing, RNA sequencing. We can look at the entire genome in some cases to see how similar one perhaps unknown organism is to a more well-known group. So these are all very quantitative. We can say that, okay, well, this species is 98% similar to species A, and it's 96% similar to species B, and it's 99.5% similar to species C. So chances are it's either species C or perhaps a very close relative of species C. Very quantitative, very relatively easy to do analyses on these types of data. We talked before about different types, shapes of bacteria. We saw these in the lab. We've got bacteria that are rod-shaped. We call those bacilli, or bacillus would be for a singular one. We have cocci in this slide over here, cocci or a coccus for just one of them. And then we have spiral bacteria over here. We're going to take a look at each one of these and kind of get a sense for what some of these different groups do. Bacilli often are involved with human disease. Rod-shaped bacteria tend to be one of the more common types associated with human pathogens. Many of these also tend to be gram-positive, and they have the ability to form endospores. And so endospores are a type of resting stage. These are produced by bacteria that have the capability of producing them under stressful conditions, Endospores can be incredibly resilient to any kind of environmental um, conditions or challenges that might show up, whether it's heat or drought or perhaps some type of chemical, lack of nutrients. Really, anything stressful can cause an organism to form endospores if it has that capability. Actinobacteria are soil bacteria. These have been important in developing different types of antibiotics. One of the main features of them is their capacity to do this. Spirochetes are spiral-shaped. Not coincidentally, they're long, sort of this spiral or helix or coil-shaped type bacteria. They may, in some cases, be human pathogens. The pathogen that causes Lyme disease, the pathogen that causes syphilis are both spiral-shaped bacteria. So, again, not all spiral-shaped bacteria are pathogenic, but we tend to know the most about those because they have a more direct bearing on our everyday lives. We can also look at how the bacteria in even a certain type, whether they're aggregating together in long chains or are they aggregating together in groups or clusters. So Streptococcus and Staphylococcus, one of the main differences between those in sort of individual cell type behavior is how they associate with other cells. One of them forms long chains of cells. The other one forms clumps of cells or groups of cells that are sort of aggregated together like we see on this picture over here on this slide here. The cell wall, we've talked about this before in plants. The main function of the cell wall in plants is to provide structure, and it's no different in bacteria. It gives them a shape and a physical boundary around them to give them some support and structure, just like it does with plants. It also gives them protection against bursting. If a bacterial cell is in a hypotonic environment, the cell wall will provide that pressure to keep water from coming in too quickly and causing that cell to burst. We've also talked about gram-positive and gram-negative bacteria in lab. Gram-positive bacteria tend to be a little more resilient in the environment at large because they are surrounded by this single thick cell wall, which gives them a fair amount of protection against outside conditions. Gram-negative bacteria are much more complex, many more cell walls, tend not to like living out in the open for too long because they don't have the same degree of protection on the outside as a gram-positive might have. So these, at least in some cases, are going to be pathogens. They need to be inside of a host or somehow protected from the environment at large to survive. One of the ways that antibiotics function is by targeting the cell wall of certain types of bacteria. So if you have, for example, a gram-negative bacteria or gram-positive bacteria, it might be possible to develop an antibiotic to attack just that type of cell wall. In general, antibiotics are best when they're fairly specific. We don't want to do a lot of collateral damage either to our own cells or even to bacterial cells that we might find helpful as part of our own body. S layer is sort of an option. Not all bacteria have this, but some of them have this layer on the outside, which gives them an extra layer of protection against the environment. All right, so I'm going to kind of take a quick look at this. The gram-positive bacteria, again, we see that up here on the left. Relatively thick cell wall. We're not going to worry about the specific chemicals involved with this cell wall, but it's a relatively thick, single-layered cell wall just outside the plasma membrane, which we see here on this slide. Gram-negative bacteria, on the other hand, have a much more complex set of layers going on here. We've got the cell membrane. We have one layer of cell. This is a peptidoglycan, as it is over here on this slide. We have a single layer of peptidoglycan, followed by some more phospholipids and proteins and lipopolysaccharides, different types of substances out here, a very complex matrix of things happening out there on the outside of that cell. So the gram-negative bacteria also tend to be those that are more frequently resistant to antibiotics, not coincidentally because of the complexity of their cell walls. This is a slide we looked at in lab showing you the gram stain procedure. Again, just to kind of review that for you, the very first thing we do with a gram stain after we fix it to the slide, cause it to adhere to the slide and dry it, is we apply our crystal violet. And after a period of time, both gram-positive cells and gram-negative cells are going to pick up that crystal violet dye. However, gram-positive bacteria are going to fix it up, or we're going to fix it much more firmly than the gram-negative will. We're then going to apply Gram's iodine, which is going to allow the crystal violet to bind to the iodine in the cell wall, if it is in the cell wall at all. It will bind there. And then we're going to take our alcohol and apply that to the slide to wash out the crystal violet that was not fixed by the iodine. So all the crystal violet that was taken up or the gram-negative bacteria were exposed to is going to be washed out. It's going to be essentially bleached out, if you like, of their cell walls, leaving them back to their original appearance. Gram-positive bacteria had their crystal violet fixed by the iodine, so they still are going to remain purple. So the very last thing we have to do, once we have our bacteria, once we have our purple gram-positives and our more or less clear gram-negatives, is to apply a second stain, which is called safranin, to stain the gram-negative bacteria a reddish or pinkish color to make them visible under a microscope. So the gram-positives are going to be purple. The gram-negatives are going to be a reddish or pinkish color. All right, so the S layer, we mentioned that a second ago. The S layer has got a variety of functions. It can be for just general environmental resistance. It can also be for adhesion. So bacteria that have an S layer are stickier. They're are able to bind to things in their environment so that a host might come and pick them up. They might be able to bind to host tissues. They might be able to bind to that piece of toast that you just dropped on the floor and picked up and blew on, thinking you might get any germs off it if you picked it up and ate it within 3 seconds or 5 seconds or 20 seconds or whatever the rule is in your house. The S layer is going to cause those bacteria to stick to whatever that substance is. And whether you blow on it or not, whether you pick it up in 1 second or 2 minutes, it doesn't matter. The bacteria, if they come into contact with that, whatever it is that you dropped, are going to be stuck to it. Capsule is another layer in some bacteria. It's outside the cell wall again. It can be used for attachment, similarly like the S layer can also do. And the capsule for bacteria that have it gives them some protection against the immune system. It makes them harder to recognize or harder to attack or harder to damage if they have a capsule than if they don't have a capsule. Bacteria that have a flagellum, we talked about this before as an example of convergent evolution. While it looks superficially very similar to a eukaryotic flagellum, it is in fact quite a bit different. It is involved for locomotion. It superficially looks the same like I mentioned a second ago, but the actual structure of it on the inside is different than what you would find with the 9+2 arrangement in a eukaryotic cell. Another option that some bacteria might have are pili. These are hairs on the outside of some especially gram-negative bacteria, which are usually there to allow them, again, to adhere or bind to surfaces or substances or tissues or something that they need to not be swept away from. Bacteria with pili are able to hang on to those substances or those structures more efficiently than those without pili. All right, so endospores, I mentioned a few minutes ago, are a resting stage that some bacteria can produce in response to an environmental stress. These endospores are incredibly difficult to get rid of. They're incredibly difficult to damage or destroy or somehow prevent from germinating. They're incredibly resistant. This is a way for bacteria to hide out. If the environment is no longer suitable, if there's a toxin, if there's a drought, if there's UV light or high temperatures or cold temperatures or just about anything, they can form these endospores and, in a sense, hang out or hide out until conditions improve. At which point the endospore will germinate, the bacteria will become metabolically active again, and they'll go back to doing what they were doing before the environment got to be too much for them. A lot of bacteria that we see as human pathogens can do this. So the bacteria that causes tetanus or lockjaw, back in the old days it was called lockjaw, caused by a soil bacterium, can be on anything that's dirty, essentially, from rusty things to things in contact with the soil. Tetanus, the bacteria that produces it, can form endospores. Anthrax is another one. Anthrax is a disease of cattle primarily, but it can be infectious to humans. It can be extremely toxic to humans. Anthrax has been one that's been looked at a lot for bioweapons, and we have a lot of concern about terrorists developing bioweapons using anthrax. If you remember back to 2001, after the World Trade Center attacks, there were some people who decided that it would be a good idea to send anthrax endospores to some politicians in Washington. And so these envelopes of white powder essentially contain endospores of these bacteria, which they were apparently hoping would germinate and cause harm to the people that they were trying to cause harm to. Botulism is a type of food poisoning. It's an extremely, extremely toxic type of food poisoning, very, very harmful type of food poisoning. And so the bacteria that produces these endospores is called Clostridium botulinum. This is an organism that you really, really don't want to have contact with, but it is everywhere in the environment. We could walk outside of your house or outside in campus, dig up some soil, and find Clostridium botulinum without a whole lot of trouble at all. The only time it's problematic is when it's ingested. And so one of the most toxic substances ever known to be produced by a living organism is the botulinum toxin. Now, I want you to think about it a second for what I just said. Botulinum toxin. Botox. Botox. Botulinum toxin. You've heard of Botox before, right? What is Botox used for? It's used for people to help rid them of the appearance of wrinkles. And essentially, how Botox works is it causes a partial and hopefully temporary paralysis of facial muscles or muscles elsewhere in the body, causing wrinkles to disappear for a period of time. So, what we have, if we take a step back and look at this objectively, is people, often without a whole lot of medical training, who are injecting the most toxic substance ever produced by a living organism into their face. Which, to me, just seems like maybe it's not the best idea. And everybody has seen people who have maybe had just a little bit too much Botox. They look a little bit like the Joker from Batman. And, it's something you might want to consider before you take that route. But this, again, is a bacterium. This is also the main one that if you buy canned goods, canned vegetables, they tell you to avoid buying the ones that have sort of a swollen or the can is sort of bulging outward in some way. If that's occurring, what's happening is the food in that can was likely contaminated by bacteria or was not cooked thoroughly. The bacteria are metabolizing, they're in anaerobic conditions, they're producing CO2 most likely, which is causing the can to expand. And if you were to buy that and consume it, it could end up giving you some very, very serious food poisoning. We've talked before about the cell membrane of prokaryotes and bacteria, and we've seen these before as being very, very highly convoluted and folded membranes. We can see in this cell over here, and especially in this one over here, all these wavy lines represent infoldings of the cell membrane, which gives these cells more surface area on which they can do their metabolism. So enzymes involved in metabolic reactions would be found in the membranes of these bacteria. Since they don't have internal organelles, they have membrane surface area to perform all the functions that they need to do. Cyanobacteria are those that are also called blue-green bacteria. They also very commonly used to be called blue-green algae, although they are not algae. They tend to associate together in colonies end to end to end to end. So they look filamentous, like algae sometimes do. These are photosynthetic. These are almost always aquatic. And they have the potential to cause a lot of harm. This picture down here on the lower right shows you the Gulf Coast of the U.S., and so in this figure, we've got a couple of things going on. First of all, we have the Mississippi River coming down right along through here and emptying into the Gulf of Mexico. And as the Mississippi River drains what is roughly a third of the landmass of North America, it's draining a lot of farmland. And there's a lot of fertilizer that runs off into the Mississippi River and eventually dumps into the Gulf of Mexico down here in Louisiana. So these blue-green algae that live in the waters around the Gulf Coast love these nutrients. They love the nitrogen, they love the phosphorus, they can't get enough of this stuff. So we get what are called algal blooms. And these algae go through the roof. Their numbers become massive and they produce more and produce more and produce more until eventually all of a sudden they all die. The end of the growing season, or if the waters become so heavily crowded with algae that they simply can't compete with each other for light anymore, these algae die, and the things that break them down decompose those algae, and those decomposers produce a lot of CO2, and they use a lot of oxygen, more importantly. So in the waters around the Gulf of Mexico, during a period of time when we have an algal bloom, it becomes very, very difficult to find fish, crustaceans, anything that's productive from a human standpoint for food or otherwise. Those organisms need to have a lot of oxygen in the water. And with the decomposers breaking down these algae and sucking all the oxygen out of the water, there's none left if you're a fish or a crustacean of some kind. So if you're a person who's a fisherman, or if you happen to own a shrimp boat, it's very difficult to find those food items that you need to harvest. So in some cases, you might need to travel a couple hundred miles off the coast of Louisiana or Texas or Alabama to find the food that you need to make a living. There are some cyanobacteria also that are involved with nitrogen fixation. So this plant up here is a leguminous plant. Things like soybeans and peanuts and alder, they are nitrogen fixers, and they have these nodules. These look like almost tumorous growths in the roots are actually produced by the plant to house the bacteria that are able to fix atmospheric nitrogen. Our atmosphere is roughly 80% nitrogen, N2. And if you remember from back in chapter 2, those nitrogens are held together by a triple bond, which is incredibly difficult to break. And one of the only things on the planet that can do it are the bacteria that live inside of these nodules. These bacteria are pretty good at what they do. They consume a lot of energy, which the plants very happily provide to the bacteria because the plants can make all kinds of glucose. They give some of that to their bacteria. The bacteria also need an anaerobic environment because to them, oxygen is toxic. So this plant very nicely provides a nice anaerobic home for the bacteria to live inside of, and both parties benefit. The plant gets nitrogen, the bacteria get their glucose and their place to live, and both parties win. Gamma bacteria are those that, again, can be pathogenic. The most famous one of these is likely E. coli, but there's a whole variety of what are called coliform bacteria, which live inside the intestines of different types of animals. Coliforms tend to live inside the guts or the intestines of mammals. E. coli is a coliform bacteria that's specific to humans. And so, of course, that one can be pathogenic, despite the fact that we need these in our guts to live a healthy life. If you're exposed to a strain of them that's pathogenic or somehow harmful, they can be extremely, extremely difficult. All right, so we've talked about some of the options here. Think about your basic base model bacteria, and once you have that in the back of your mind, it's got a cell membrane, it's got some cytoplasm, it's got a chromosome, it's got a cell wall. Once you have that, you can start adding on some other features, like perhaps a flagellum, or some pili, or a capsule, or an S layer, or endospores. Lots of other kinds of things which can help the bacteria make a living. We talked about this, I think, a little bit before, how ribosomes between prokaryotes and eukaryotes are also different. We talked about prokaryotes having a 70S ribosome, eukaryotes having an 80S ribosome. Some antibiotics work on this basis, and they're able to fight off cells or kill cells that have the 70S type ribosome, which is great because it allows the antibiotics to target just the bacteria and not your cells, which again is what you want. When you're taking an antibiotic, you want it to kill the pathogen. You don't probably want it to kill your own cells. So we know about the nucleoid region. They, of course, have no nucleus. The name prokaryote means no nucleus or before nucleus. So they have no nucleus, but they do have a place in the middle of the cell where we can very predictably find bits of DNA. This, the chromosome, perhaps also plasmids. Plasmids are bits of DNA that are not essential for the life of the bacteria. They're like extra bits of DNA that perhaps the bacteria has picked up at some point during its life. They can be expressed, they can be used, they can be turned into proteins, but they can also be taken away experimentally and the bacteria will function just fine. So bacteria have that single circular chromosome, but they may also have these plasmids, which is sort of like a secondary or another set of DNA which they can use for different purposes in their lives. Alright, so we have Staphylococcus down here on this bottom picture, Streptococcus up at the top. Streptococcus form these nice long strips or lines of bacteria, whereas Staphylococcus tends to form together in clumps. Notice how they're both cocci, they're both round-shaped bacteria, and also notice the scale here. This line right here represents 2 micrometers. So every single one of these cells might be, what? We're looking at maybe a half or maybe a little bit more than a half of a micron across. Very, very, very tiny. Alright, so we're going to cut off this section of the lecture now. I'm going to restart a second section. I don't want these files to get too big, so I'm going to stop this, cut this off, and I will start part 2 of this wonderful lecture.